Hello and welcome to the Executive Report for Natural Killer and NK Cell Therapies. Today we'll cover the key industry drivers, competitive landscape, top companies, and our outlook for this dynamic space. 1. Natural killer and NK cell therapies represent a rapidly advancing domain within cancer immunotherapy. Unlike CAR T-cell therapies, NK cells offer innate immune surveillance capabilities that are less dependent on tumor-specific antigens. Their potential as off-the-shelf treatments enables broader accessibility, faster manufacturing timelines, and a more favorable safety profile. As the field matures, NK cell therapies are emerging as serious contenders or complementary alternatives to CAR-Ts, particularly in hematologic malignancies and selected solid tumors. 2. Market landscape. To point 1 industry overview. The global NK cell therapy market was valued at approximately $2.3 billion in 2023 and is expected to reach nearly $9.8 billion by 2030. This growth corresponds to a compound annual growth rate CAGR of approximately 25 to 28%. The clinical focus spans both hematologic malignancies, such as acute myeloid leukemia AML and non-Hodgkin lymphoma and HL, and solid tumors including lung, liver, and breast cancers. There is also exploratory potential in infectious diseases. To point to growth drivers, several factors are driving interest and investment in NK cell therapy. The ability to create allogeneic, off-the-shelf products leads to shorter turnaround times and lower costs. NK cells present a more favorable safety profile, with a significantly reduced risk of cytokine release syndrome (CRS) and neurotoxicity compared to CAR T therapies. Advancements in genetic engineering, cell expansion, and cryopreservation have made manufacturing more feasible. NK therapies can be combined with immune checkpoint inhibitors or monoclonal antibodies for enhanced efficacy. To point 3 challenge. Despite the promise, NK cell therapies face several challenges. NK cells tend to have limited persistence and expansion in vivo, which can reduce therapeutic durability. Manufacturing at scale, especially with consistent potency and stability during cryopreservation, remains complex. Regulatory pathways are still developing, and there is limited precedent compared to the more mature CAR T space. 3. Value proposition and strategic. NK cell therapies provide a unique value proposition relative to CAR T therapies. Firstly, they can be sourced from allogeneic donors, induced pluripotent stem cells (iPSCs), umbilical cord blood (UCB), or NK cell lines. This contrasts with CAR T therapies, which are often derived from autologous T cells, necessitating a lengthy and patient-specific manufacturing process. NK therapies can be produced and delivered within days to weeks, whereas CAR T therapies may take 3 to 5 weeks. This faster timeline improves clinical responsiveness and reduces patient attrition. 4. Top 3 companies in the NK cell therapy space. For point 1, Therapeutics. Therapeutics is a leading developer of iPSC-derived NK and CAR-NK cell therapies. Its lead candidate, FT516, is an iPSC-derived NK cell product currently in clinical development. The pipeline also includes FT538, an enhanced CAR-NK product, and FT576, targeting multiple myeloma. Fate's iPSC platform enables clonal, genetically defined, and scalable production of NK cells. The company has a significant partnership with Ono Pharmaceutical and has demonstrated multiplex engineering capability. For point, Nkarta Therapeutics. Nkarta focuses on CRISPR-engineered, off-the-shelf NK cell therapies. Its lead candidates include NKX101, which uses an NKG2D activation receptor, and NKX019, a CD19-targeted CAR-NK product. The pipeline is particularly focused on AML and B-cell malignancies. Nkarta has built in-house manufacturing capabilities and uses CRISPR modifications to enhance persistence and cytotoxicity. For point 3, Artiva Biotherapeutics already develops cryopreserved, allogeneic NK cell products designed for antibody-dependent cell-mediated cytotoxicity (ADCC). Its lead candidate, AB101, is being studied in combination with rituximab for lymphomas. Artiva also has 𝗖𝗔𝗥-𝗧 and EGFR targeting NK products in its pipeline. The company has strategic collaborations with Merck and GC Labtech and emphasizes scalable, cGMP-compliant manufacturing processes. 5. Due diligence framework for NK cell therapy investments. A robust due diligence approach for NK therapies should include scientific and technical validation, examine the persistence, cytotoxicity, and expansion efficiency of NK cells. Assess innovations in targeting mechanisms and immune evasion strategies. Clinical data focus on early-phase trial results, particularly objective response rates, duration of response, and safety profiles including CRS, GVHD, and other immune-related toxicities. Manufacturing feasibility. Review the scalability of the GMP process, stability of cryopreserved products, and the source of NK cells iPSC, donor-derived or UCB. Intellectual property. Assess the breadth and enforceability of IP related to gene editing, CAR constructs, IL-15 expression, and proprietary cell sources. Regulatory momentum. Check for regulatory designations like IND acceptance, RMAT, or Fast Track, and assess the risk profile of pre-IND and IND-stage candidates. Partnership ecosystem. Evaluate partnerships with pharma, biotech, and CDMOs, as well as internal capabilities for manufacturing and commercialization. Commercial outlook. Understand the reimbursement landscape and value-based pricing potential. Analyze how the therapy competes with or complements CAR-T options in specific indications. 6. Strategic comparison. NK versus CAR-T cell therapy. While CAR-T therapies have a longer clinical track record and multiple FDA-approved products, NK cell therapies are gaining ground due to their off-the-shelf potential, reduced toxicity, and cost-effectiveness. CAR-T therapies are largely autologous and patient-specific, requiring weeks for manufacturing. Their long-term persistence and durability is a strength, but also a potential liability if associated with chronic toxicities. Additionally, the high cost, often exceeding $400,000 per treatment, poses a barrier to broader access. In contrast, NK therapies can be manufactured in advance from universal donors, or iPSCs, and stored cryogenically for rapid deployment. Their innate immune function reduces dependency on tumor antigens and lowers the risk of immune-related toxicities. While their persistence is generally shorter, efforts are underway to enhance longevity through IL-15 transgene expression or checkpoint inhibition. Scalability is a major advantage for NK therapies, making them suitable for outpatient settings and broader commercial adoption if efficacy continues to improve. 7. Conclusion and recommendations. Cell therapies offer a compelling alternative or complement to CAR-T therapies. Their potential to deliver effective, safer, and scalable treatments at a lower cost makes them particularly suitable for indications where CAR-T is impractical due to cost, time, or toxicity. iPSC-derived platforms in particular may define the next generation of cell therapy manufacturing. Thank you for watching. If you enjoyed the video, please consider giving it a like and subscribing for more content. Until next time.